Welcome to Therapeutic Momentum. We take you on an inspiring journey, spanning breakthroughs in science and medicine development. Over the last century, our average life expectancy has increased by more than 20 years. Join us as we go behind the scenes with medical researchers, clinical developers, and other experts working together in the race to tackle life-altering diseases with new treatments and cures.
I'm Karen Akinsanya. I'm a translational scientist working at the intersection between biomedical research and emerging technology. Thanks for tuning in to Therapeutic Momentum. Let's get started. I'm delighted to welcome Rupert Vessey to the podcast. Rupert has served in C-suite and senior leadership roles at BMS, Celgene, Merck, and GSK, where he's ushered numerous products through discovery and development. Most recently, he joined Flagship Pioneering as Chief Scientist and Executive Partner. Throughout his impressive journey, Rupert has nurtured the careers of scores of scientists, and he's a highly regarded role model to many, many more. A very warm welcome, Rupert, and thanks for joining us.
Thank you, Karen.
So, first of all, Rupert, I'd love to hear more about your journey. Obviously, you've reached the very pinnacle of pharmaceutical research leadership, but perhaps you can take us back to the very beginning, how this all began, what your ambitions and aspirations were, any interesting pivots along the way, and any reflections that you have.
Yeah, let me do that. So I think I really got interested in biomedical science, I would say, when I was in high school. But there had been a lot of influences on me, like there are on others from my family. So in particular, I would say my father, who is a physician and a scientist. He was an epidemiologist and just a great role model in many ways. In fact, he tried to stop me from becoming a physician because he said it was a really difficult job and I should think about doing something else. But besides him, there were others as well. In particular, my maternal grandfather, who was a remarkable person who I only knew briefly because he died when I was young, but his story was pretty remarkable because he grew up in pretty tough circumstances in Leeds in the UK, and through an anonymous donor was able to finish at the grammar school in Leeds rather than going to work in his father's shoe repair shop. And he became a physician and he did a PhD in biochemistry. And before World War II, he was involved in clinical trials administering the first ever purified vitamin B to patients with vitamin B deficiency. He also set up a research center for the UK Medical Research Council in the Gambia, so spent many years in both Africa and in China. It's a regret to me that I didn't know him better, but he's always sort of been there in the back of my mind as an inspiration. So I think, the seeds were sown. And then I remember when I was what we call a sixth former in the UK, we had a fantastic biology teacher called Dr. Tim King. And we were very, very lucky to have him. But it was at a time when molecular biology was just beginning to be applied to biotechnology. So I remember him teaching us about the production of human insulin through recombinant DNA technology. And I just thought that was, incredibly exciting. So I wanted to go to university to study medicine, but also to do basic science as well. And so I was lucky enough to get into Oxford, and I went to Merton College there, which was a great place, and did my undergraduate degree. We had some great tutors, some of whom were also very inspiring people. And then I went to the clinical school, and when I finished the clinical training, I went into my initial junior doctor jobs. Of course, Karen, you'll remember this is where I first met you, actually, because
Yes.
One of the jobs I had was at the Hammersmith Hospital working in the endocrine group for Professor Sir Stephen Bloom, who's near and dear to both of us as a mentor. So he gave me opportunities to work in the lab, even though I was supposed to be working on the ward. And he's somebody who has also been an inspiration and a supporter throughout my career. Then I went back to work in Oxford. I got an MRC training fellowship, and I went to work in what was called the Weatherall Institute of Molecular Medicine, named after David Weatherall, who was another fantastic physician scientist who was Regius Professor of Medicine at the time. And I worked in John Bell's lab there. And John is also a remarkable person. He's just retired as Regius Professor of Medicine at Oxford, actually, after essentially transforming the entire biomedical community in that institution and keeping it at the very forefront of biomedical science. So immunology was a great discipline in Oxford. There was a whole bunch of really good immunologists, Andrew McMichael, Alan Townsend, John himself, amongst others. And so that was a great place to learn immunology. And I finished up my DPhil PhD equivalent, and I really enjoyed doing that. So now I was qualified in medicine, and I had worked in the lab. And the next thing looked like a career in academic medicine in the UK.
Right.
And so I became a clinical lecturer at St. Bartholomew's Hospital in London. And I just found that the freedom and ability to self-determine that I had had in the lab in Oxford was hard to reproduce. Mm. And so I got to a kind of pivotal point where, I was trying to decide whether to really pursue this academic medical career or whether to think about applying the things that I had learned in a different way. And one day I was sitting in the accident and emergency department on call, and it must have been late at night because there wasn't too much going on at that point. And I looked in the BMJ, British Medical Journal. In those days, they still advertised jobs in print. So you have to understand there's no LinkedIn or anything like that.
Yes, no computers.
Exactly. So I saw this advertisement at what was then Glaxo Wellcome, who had just invested in 2 therapeutic vaccine platforms. One was a DNA vaccine platform with a company called PowderJect, and the other one was an HSV vaccine that had come out of Tony Minson's lab in Cambridge, which was replication deficient, so would go through a single round of replication, enough to really stimulate an immune response, but not to cause an infection.
Right.
I thought these things were fascinating, and the job asked for a physician with, PhD in molecular immunology. And I thought, I'm going to investigate this. Not many people with a clinical and PhD background moved out of academic medicine into industry in those days. It was a sort of heretical thing to do. So I took myself off to Glaxo Wellcome one afternoon without really telling anybody. But when I got there, I found that I interviewed with just an amazing group of people. People at the forefront of population PK science for HIV drug development, and in particular, the head of what was then the clinical pharmacology department, Dr. Garth Rappaport. And I had known Garth briefly when he was a registrar in Oxford. So I was quite inspired to meet him, and he was very entrepreneurial and very visionary in terms of what he wanted physician scientists to do in Glaxo Wellcome. In those days, research and development were completely separate disciplines. Research went on in a lab somewhere up in Hertfordshire. Development went on in a building somewhere in Greenford. And, occasionally, or hopefully more than occasionally, molecules would appear. They would go into development, and then development would sort of do something with them and hope they turned into drugs. And he really wanted people who can live in both worlds. And so that was just sort of tailor-made for me. And, I loved it. The projects that I worked on, the 2 vaccine projects, actually were both immunogenic. And, in some ways, the DNA vaccine in particular is a sort of forerunner of where we are today with nucleic acid vaccines. But I also worked on some other very interesting projects in inflammation, monoclonal antibody projects, and various other things. So that was a great opportunity.
It must have been incredibly exciting. At that time, those were the early days of monoclonal antibodies, which are really now a significant portion of the drugs that patients have access to. You had this very early translational experience at GSK in the UK. How did you end up embarking on a career in the US?
one day I was just sitting in my office in Greenford and my phone rang, and some person from a place called North Wales, Pennsylvania, called Tony Fischetti, who's a recruiter, he's still a recruiter, rang me up and he said, "Oh, we saw your name on a poster, and Merck is looking for people who know viral immunology. They have a vaccine portfolio. You look like you'd be a good person for this. Are you interested?" And I thought about it for a bit. And although I loved my job at Glaxo Wellcome, I thought, well, this is an opportunity because you know as well as I do, the biopharmaceutical industry is pretty big in the United States. There's a lot of opportunities. So I thought, well, I'll investigate this. And I did. I flew over a couple of times, met with all sorts of different people at Merck, including Barry Gertz, who at the time was running their early clinical development, who went on to be, the head of all development at Merck. I think Roger Perlmutter was actually Merck at the time running research. And the vaccine research was run by Emilio Amini, who was a fantastic infectious disease and vaccine researcher. So they were really inspiring people. So they made me an offer and I said, I'll take it.
We overlapped at the Hammersmith Hospital with Steve, but I had no idea that you and I both received phone calls from Merck while in other roles. I flew for Ferring Pharmaceuticals to North Wales, Pennsylvania to meet Gail Murphy in clinical pharmacology, and then to Rahway to meet Dan Bloomfield, Keith Gottesdiener, Nancy Thornbury and Anne Webber. I loved the idea of pursuing molecules from discovery to the clinic, and that's what brought me to Merck.
Now, since I worked in the UK, Glaxo Wellcome said that's fine, but we have a policy of 3 months of gardening leave. So I had to sit at home in London for 3 months. And by the time I showed up at Merck, I think they'd forgotten that I was coming. I had no computer ready. And they're like, "Oh yeah, that's right. We did hire," because they're used to people coming in 2 weeks in the US, but it was a different arrangement. Anyway, I moved to Pennsylvania and started working on the vaccine programs there, which was great. I got some really good experience in sort of later phase development, working on varicella vaccine and also Zostavax, which was the first shingles vaccine. It's subsequently been supplanted by Shingrix. But at the time, it was the first one. And we did a lot of T-cell biology, trying to understand correlates of protection and so on, which is a hard thing to do in large-scale vaccine trials.
You'd learned a lot about vaccines development at Merck and seemed to be enjoying the work environment. So what was it that drew you back to GSK?
During that period of time, Glaxo and SmithKline had merged. And so now they had operations in Pennsylvania. Garth Rappaport, who I'd worked for before, called me up and said, "Now you're over there. We've got all our respiratory and inflammation stuff is partly in the UK, but a lot of it's down there in Upper Merion, Pennsylvania. What do you think about coming in and doing some work for us there?" And Tachi Yamada, who was the head of R&D, had come up with what I thought was a really interesting structure and way to run R&D. So he had taken this concept of physician scientists working across the research and early development divide and really codified that into his organizational design. So he created these CEDs, or Centers of Excellence in Drug Discovery, that did everything from targets through drug discovery through to some designated proof of concept point. And they were great. They were units of about 300 people. They had very strong entrepreneurial leadership. They had their own budgets. They were really driven to take these molecules to this point. And then there was a later phase organization, the Medicines Development Centers, that took them and did the confirmatory trials and the life cycle management and regulatory approvals and all the rest of it. And I found that to be a really, really dynamic environment to work in. It was great to work with the researchers. You had a really clear mandate. And Tachi Yamada, who, as you know, sadly passed away a couple of years ago, was a fantastic inspiration. He was a great leader. He had very strong medical principles as well as being a really good scientist. Actually, even through the remainder of my career, I used to meet with him every year at JP Morgan, and he was just a wonderful person to know. I think we all really miss him.
So really translational science, the idea of reaching in both directions.
That really was my passion, to do that and to see those ideas go from research and then have the first steps into clinical development and then see the signals starting to emerge and think, okay, we've got something here that, has a chance of working. I had a great team there, and Garth was also a tremendous sponsor for me. He built me opportunities through the next 5 or 6 years while I was there to grow. Then, as things so often do in industry, there were significant management changes. So Tenchi went to run the Gates Foundation, and that changed the organization somewhat. And, during that period of time, interestingly, Merck had evolved their organizational structure to look a bit more like what, GSK was doing. So that was really Peter Kim's concept to create these franchises. And I initially got a call from Barry Gertz at Merck who said, we'd like you to come back. And the job he actually offered me was in later development, interestingly.
I see.
Working closely with Alan Ezekowitz, who ran the respiratory and inflammation franchise. So I took that position. I'd been there for about 3 months. And I got a call from Peter Kim. And he said, well, here's the real reason we wanted you to come and work here, because you've worked in this translational environment, and we think that that's what you should be doing here. But what we actually want you to do is move your family from Pennsylvania to Seattle. And we want you to be the site head and leader of Rosetta Informatics, which Peter had led the acquisition of that a couple of years before. He said that there are amazing people there, incredible technology, but we need it to be connected into, what we're trying to do here in terms of drug discovery. So Laura, my wife, and I and our kids were a little bit, 4 kids.
Right.
And 3 of them were in school. So we obviously looked at this with a little bit of trepidation. And Peter said, oh, you have to do this. This is the best job in the whole company, he said. So actually it was Laura who really convinced me to take that job. She said, I really think you should do this and we'll make it work. She also worked for Merck at the time, and it meant she had to do her East Coast job from the West Coast, which was, quite a difficult thing for her to do. But we moved out there, and this was really a sort of transformational moment in my journey, because I went from somebody who was used to running things that I knew about, supervising people where I could actually have done their jobs for them, but I was guiding them and teaching them. And, I had groups of, 20 or 30 people. I knew all of them really, really well. To suddenly I had this whole site with 300 people in it. And, I probably couldn't have done the jobs of anybody. We had brilliant computational scientists like Eric Schatt. We had Peter Lindsley, another great scientist, ran a lab doing lentiviral target validation. We had a software business. And the gene expression lab. So we developed and sold software to be able to look at gene expression, because these were the early days of molecular profiling, and a gene expression lab that could profile, thousands and thousands of samples a year. We also had one of the 10 biggest computer clusters, because there was no cloud. All the computers were in the top of the building.
I see.
In air-conditioned space. What did I know about linking hundreds of servers together in a building? So it was kind of intimidating in a way, but you had to move from sort of trying to be the expert in everything to somebody who made connections, saw the bigger picture, guided people to try to do things that were the best value, and leading people in a way that really wasn't about yourself, but was about everybody else. And so I really think that through that experience, I changed a lot in the way that I thought about things and the way that that I approached things. So that was kind of a big moment.
It's truly amazing to look back and realize how pivotal that position was in terms of your leadership journey. I can relate to this, having left Merck, where there were established groups to join Schrödinger, a very well-established software company, to try building drug discovery and development in-house almost from scratch. So Rupert, you're site head at Rosetta on the West Coast, and you've learned a lot about how to interweave technology into the early stages of drug discovery. What happens next?
Unfortunately, Merck was going through significant reorganization, and there were a lot of site consolidations that took place. And the site in Seattle, the Rosetta site, was a site that the company decided that it couldn't keep in its current form, so we had to close that. That was a hugely painful experience, because by that time I had really grown fond of the people who worked there and had a very strong belief in what we were trying to do. Stephen Friend, who had founded Rosetta, was still working for Merck, and he came with me to deliver this news, for which I will be forever grateful. I thought that was an act of extraordinary generosity and kindness on his part to support me. So I learned a lesson there as well. Anyway, we went through that, but we sold the software and data business to Microsoft, and some people went with that. So insofar as we could, we made the best out of that. And I was helped a lot by the chief operating officer at the time, who was a chap called Doug Bassett, who's worked with me subsequently. Really an amazing person to go through that with. So that happened, and then Peter Kim called me up again, and
the infamous call.
He said, I've got another assignment for you. And I said, oh yeah. And he said, well, this one really is the best job in the company. And I said, I think I've heard that before. He said, well, I want you to move to Boston and run the Merck Boston site. And that was a fantastic opportunity. The site had not been fully populated at that time. It was a place where we had transferred a lot of the informatics skill sets from Rosetta, probably about 80 to 100 people. Merck Frost was another site that was going away in the network reorganization, but
a lot of talent there.
A lot of great talent. And Kathleen Metters, who ran basic research at the time, was able to help me get a lot of great people to move down. And so over the next several years, we created what I thought was just a really vibrant culture, a great site that really started to be able to deliver on its full promise. And I think we just got that to the place where it needed to be. And then the Merck Schering-Plough acquisition came along, and I got another assignment because Peter Kim called me up again and said, I know you love it up there in Boston, but I need you to come and help me with the integration of Kenilworth into the company. So Laura and I undertook our fifth move in however many years, and we moved back down to New Jersey and took on that responsibility.
But that's incredible. I mean, it does say something about you and indeed your family, that you were able to make these pivots, changes, change in location, change in jobs, change in scope of responsibility so many times. What do you, when you reflect on that, do you think it takes a special kind of person?
I think it probably does take a high degree of resilience. And the stresses placed on families should not be, underestimated. Yeah. Sometimes we look back on it and wonder how on earth we did it. My middle daughter works for Nestle, and she just moved to Switzerland. And she found that to be very challenging, although she chose to do it. But she said to me, I think the reason I've managed to do this is because of all the moves we did when I was a teenager, because I built up the ability to get through this. So, maybe there was some silver lining in there. But it's not that common in the research organization to make, commercial people do this a lot. But, and, Merck obviously helped a lot, making things easier in terms of moving. But it was challenging. So I spent the next sort of, period of time in New Jersey, and we got the Kenilworth thing sorted out. And then I ran, the respiratory and inflammation franchise for a while. And then Peter retired from Merck, and Roger Perlmutter came back as head of R&D. And for a couple of years, I ran discovery and early development with Roger. That was a really fantastic learning experience. Roger he's a unique individual. And I learned a great deal from the period of time I spent working with him. I still sometimes, in my more recent jobs, I would sit in meetings and think to myself, what would Roger Perlmutter do in this circumstance? And clarity and decisiveness and insight.
Yes, agreed. I had the opportunity, as you know, to join business development after being in the labs and in clinical at Merck, and had a chance to work with Roger. And you're quite right. I mean, he has an incredible ability to synthesize vast amounts of information. And so you're right to point out that decision-making is really one of the most important aspects of leadership in R&D. Roger, and certainly you and others, and something I have to do a lot more of now in my new role, really helping teams, helping large organizations make decisions that we believe will be best for the pipeline and the programs.
It's amazing as we go through this, I think about the people who I've encountered along the way. But I got a call from a scientist called Professor Sir Mark Feldman. So Mark is a Lasker Prize winner because he really was the person who unlocked the use of TNF therapy in rheumatoid arthritis. And I had gotten to know him. We were quite friendly. We had a number of collaborations and things. And he said, I want to put you in touch with another friend called Tom Daniel, who runs research and early development at this company called Celgene. I knew Celgene, but I didn't know that much about it. But I went up and I had dinner with Tom, and then Bob Hugin, who was the CEO at the time, joined us. And we just really hit it off. We really enjoyed meeting. Philosophically, we had like-mindedness, and I was really impressed with the way that Celgene approached a blend of internal capability and external science. I got to meet George Golombesky as well and listen to his vision, and I thought, well, I'd been at Merck for quite a long time at that point. I thought, well, this is probably time for me to do something different. And this feels great because unlike transplanting into another huge organization where I have no history, here I will be able to get to know the 400 or 500 people in the research and early development organization very quickly. And I'm going to learn so much about collaborating in the biotech ecosystem because of such a unique way of doing this.
Right. And by that time, the Rosetta experience, the Boston site, you'd really sort of mastered sort of coming in as a new leader into an organization of a certain size.
Of a certain size,
yes.
Exactly. And so that just really felt like a great fit. So I went in and I worked with Tom for about a year. And then, he said, look, it's time for me to retire and I want you to take over. I didn't know that at the time. It was just great. From Tom, I was lucky enough to inherit some fabulous people, Kristen Heggie, who did so much for cell therapy, as just one example, but there were numerous other great people on my team. And then I added some others, some of whom you know well, like Richard Hargreaves came in to do neuroscience. Robert Plenge, of course, came in to do the immunology side of things. And Doug Bassett, who I'd known at Rosetta, came over to start building up the informatics side of things, which we didn't have much there, but we knew how to do that. So we set about developing that organization, and we did that for about 5 years or so. And then BMS acquired Celgene. That was exciting in some ways. I mean, after the BMS acquisition of Celgene, I think in the next 3 years, there were 9 or 10 new products approved, and 6 or 7 of them all came from the efforts that Tom and then my team had made at Celgene, and some of them are very novel and differentiated. So that was exciting. I chose to stay at BMS. I mean, it was a decision to be made at the time. I knew it meant moving back to a bigger company setting. But on the other hand, I think Giovanni Caforio, who was until recently the CEO of BMS, he wanted to try to create a research and early development organization that was different from the one BMS had. And so keeping me meant that he was able to keep most of my team. We lost very few people. We were able to really diversify the site footprint, bring in important new modalities to BMS, in particular protein degradation and cell therapy, to augment some of the very strong traditional modalities in medchem and biologics discovery, and really create a broader portfolio and a bigger set of opportunities. And so that brings me pretty much all the way up till now. I was really lucky to be able to bring Robert Plenge with me to BMS, and he had been a planned successor. I think it's not always that you get to be able to hand over to the person that you've worked with for most of 10 years and developed to a certain point. And I think that's a good feeling, because you know that that person will build on what you've done, but will also recognize the value of what has been done before. I have a photograph of Tom Daniel and me and Robert Plenge. And if you add all of that up, and Robert does what I hope will be a few good years at BMS, it'll be 20 plus years of continuity in terms of research. And as you know, research takes time. You have to keep plugging away at things. Obviously, you have to stop things and keep things going, but you need to have perseverance in the things that you're good at. And companies go through waves of innovation and then consolidation, and that's the way that this goes. So I think having some of that institutional knowledge that you don't lose over time is really valuable to larger companies. And so then, I'd known Nubar for 20 years, and we talked on and off. And Flagship Pioneering is an amazing organization. I mean, what Nubar has built is incredible. So when he offered me the chance to join him as chief scientist, I jumped on that. It keeps me more than up to date. I get to work with amazing people here who are incredibly entrepreneurial, and I'm learning not only scientifically but also on the business side as well, because it's obviously a very different setup from the one that I grew up in.
Yes. Yeah. Well, what a terrific journey. You touched on a few things there that I think are worth going back to. So obviously you started with your medical training and sort of one foot in academia. You obviously ultimately transitioned into industry. I want to return to the sort of idea of industry and academia, because I think another feature of your career has been obviously nurturing organizations, building organizations, but also reaching outside of those organizations and collaborating, whether it be with biotech. I think you mentioned Celgene, that had a very strong model around collaborations with biotech. But I think also academia has featured in terms of how you thought about running research.
Yeah.
So maybe you can talk about that a bit.
Karen, I think that's a really interesting topic, and one that we could probably spend a long time on, because I've seen a lot of different ways of working with academia, and some of them have been great and some of them have not been so great. But I think the key thing, and this sounds sort of obvious in a way, but it has to be a real collaboration. So there has to be something from both parties that is coming to the table that's going to make the science that you're doing better. And that drives enthusiasm. And of course, that's the basis of a great collaboration. And we've had some really good ones. I mean, one I'm really proud of is something that we set up in Oxford with Tom Daniel and Mark Feldman, which is now into its 10th year, I think, where we established a translational fellowship. But it wasn't just about providing funding. It was about providing opportunities. So, and the idea here really was Mark's, and he wanted to give physician scientists in particular, but also PhD scientists, an opportunity to understand what translational science means in the industrial setting.
Yeah.
So we had, 4 or 5 fellows a year who we funded to do shared projects where they had a supervisor within the company and one within the academic institution. And the projects were really centered around understanding drug mechanism of action, or patient populations that were relevant to a particular therapy. The very first fellow we had actually was instrumental in working out the mechanism of action of IDFA. And her work went into the regulatory submission for IDFA. And every time we used to kick the program off, I always used to use that as an example and say, this is what we expect from you. I was joking. But, that was a sort of remarkable start. So I think that sort of thing is really important. And then incubating things within academic institutions. There's obviously fantastic ideas, but you need to find ways to bring the right capabilities to the table so that you get really robust assay systems. You're using the right kinds of compounds to validate things so that the data that you generate is something that you really feel you or others can make an investment in and stand behind.
Yeah, there's this sort of perhaps misconception that industry comes and sort of grabs amazing science and sort of takes it away into the sort of company and doesn't look back. But I think really the truth is that it is about bringing the skill set of industrial pharmaceutical researchers, the rigor, the sort of validation, the understanding of PK/PD, and sort of bringing that into the academic environment.
Well, there are fantastic scientists in all the companies I've worked in. There have been amazing, talented scientists who have a very sort of applied bent to what they do.
Yes.
And so I don't think it's about intellectual skills or scientific skills, that these things are very equal, but it's about how you focus those things. And they're actually highly complementary.
Indeed.
And when it works, it's really great. And we've had many examples of that. I mean, the sort of thing that doesn't work very well is, just sort of funding something and then hoping that you'll get something out the other end of it. I mean, that's you can do that kind of thing to help the ecosystem in a general way, but that's not a collaboration. That's a different way of doing it.
Yeah, the arm's length relationships tend not to work so well.
Yeah.
So, I mean, over the course of your career now, I think something else has been happening, which is the really growth and emergence of biotech. And you already mentioned the work you did with BMS and Celgene to bring forward new modalities. I think we're sort of in a sort of interesting moment as both now tech comes into biopharma research, but before tech, there was molecular biology, as you said, really amazing leaps forward in terms of innovation. So, maybe you can reflect on the interactions you've had with biotech, the innovation that you've been seeing sort of come forward as additional modalities of helping really cure diseases at this point.
Yeah, no, it's been an amazing period of time. Sometimes I like to reflect back on being a sort of final year medical student and taking my exams and thinking about the patients that we saw in the clinical exams. And actually, I once made a slide of a whole list of diseases. It could be HIV, HCV, rheumatoid arthritis, multiple sclerosis, psoriasis, myeloma, melanoma, the list goes on. And then you look at what the outlook is for people who have those diseases today and the innovations that have taken place in those 25 years. And I know that medical students just don't see the same morbidities that I would have seen just a quarter of a century ago. And that is because of innovation, a lot of which has taken place in biotech companies, but also been polished and developed and enhanced within large companies where you have scale and other types of capabilities that allow you to take those things forward or even understand how they work better than maybe the innovator company originally did. Yeah, there's just been, so much. I mean, one thing I've really learned over this time is that new modalities take quite a long time to become useful. And the first iterations of almost everything, I mean, whether it be monoclonal antibodies, cell therapies, RNA therapeutics, we could think of other things as well, gene therapies.
Yeah.
The first attempts often don't work very well, or they struggle to be commercially viable because manufacturing or whatever hasn't been perfected. But provided they demonstrate some value and promise, our industry is amazingly good at then taking those things and refining them and making them better and better and better until the point where they become really well-established modalities that you can turn to.
Yeah, I remember perhaps, 21 years ago, Merck actually acquired Cerner.
Yep, that's right.
And I think in the end spun it out.
Yeah, back to Alnylam. Yes.
That's right. And now we have our first approved product. So
right.
that sort of cycle,
yes.
20, maybe 30 years.
The cycle is long, and cell therapy is the same thing. I mean, Kristen Heggie, who I mentioned earlier, spent 25 years of her career in the biotech. Her first project was in cell therapy, and her last one was the approval of Abecma. Yeah. And through all that time, she worked on other things as well, but she worked on that, and she's written some really nice retrospectives on her journey. So you have to have patience. That's one of the things that's great about Nubar and flagship pioneering, actually, and Moderna is a prime example. Yeah. Look for new modalities, set up the platforms, find the right application, but stay in it. Not every time, but many times the right time will arrive if you're patient.
Well, this whole space, drug development, technology development, innovation, new modalities, it requires an enormous amount of patience. When you look back at some of the products that you've worked on, I think we hear these numbers that are put out into the public, billions of dollars to sort of fund research, 10 years to develop a product. When you look back at some of the products you've been involved in, are there moments that you look back and say, yeah, we really cracked that one? It took time, but we cracked it.
Well, I think some of the clinical data that we got working on the cell therapy products certainly felt that way, because you could have really profound big effects in small patient numbers, and now you have real life-saving products. But this is, again, a modality that needs to be further improved, and people are working really hard to do that. I was lucky enough to be at Merck as the checkpoint inhibitor, story began to unfold. And although I wasn't directly involved in the project, parts of my group worked on it, particularly around the biomarkers and some of the early clinical trials. But watching those data unfold was pretty remarkable. Yeah. That was definitely an exciting time. But there's lots of things that haven't worked, of course. Yeah. And getting to that decision quickly with really robust data that you can learn from, that's one of the most important jobs of a translational person in industry. Yeah. The worst thing is when you spend a lot of money doing something and you learn nothing. That's really, really a bad outcome, because it often leads people to just do the same thing all over again, and you waste a whole load more time and money.
Yes, yeah. So resilience is a word that's come up. There is, I think, a special sort of personality that can do the moves that you've described, traverse early and late, also work in all sorts of different environments. What sort of keeps you going? What are the things that motivate you? Are there things that we wouldn't naturally think of about Rupert that you want to share with folks?
Well, I think a lot of them are the same things that would motivate anybody who works in our industry, and that is really the excitement of a project and looking forward to seeing the data come out. And for me, it was especially the initial clinical data, because that's really where the action starts. So you've done all this hard work, and now you're going to do this experiment. And every time we got something that gave us a positive signal that we could advance, that was super exciting. It was also great to get an experiment that was really, really clean and definitive, where we knew we'd properly tested the idea and helped the field. So there's that angle. But, as I advanced in my career, particularly after the, time I spent at Rosetta, the thing that really came to motivate me a lot actually was being able to try to provide an environment where the scientists in the organization could do really fantastic things, just to get as many roadblocks out of the way, to give them creative space so that they could have ideas and we could pursue those ideas, and then hearing about those things in informal settings, not committees to review them, but just to learn about what they were doing and feeling their excitement about the work that they were doing. You become more of a sort of coach in that sense, in that you're trying to get a group of people to succeed. And it's not about you or yours, it's about, their successes. And that was a very powerful motivator for me for a long time, and actually still is here at Flagship. When our ecosystem companies are making progress and things are going well, I'm not doing that work. Right. I'm trying to help them when they need input from me, whatever that may be. But seeing them so enthused about what they're doing is really exciting and fun. And so that's a big motivator for me.
And that goes back to your Rosetta days. I think you described it as perhaps the first time where you had to work with and through others as opposed to actually generating the data yourself, and finding a way to actually motivate and make that space for people is a huge part of leadership, isn't it?
For me, that is what leading people is all about.
Yeah.
It's about moving to a state of a more selfless state of mind and not trying to take credit for everything that happens, but taking joy in other people's successes is just incredibly important. I think if you can't sort of get there, then you're going to struggle to lead large groups of people, really.
Yeah. Yeah. Any favorite books, any favorite music that you want to?
Well, books, I jotted a couple down, and I love to read, so there are lots of books, but I remember reading The Selfish Gene the first time, and I'm sure many people read that, and I just thought that was kind of fascinating. And I do enjoy reading books like that. There's another book called The Sports Gene, which is also interesting. I'm not one for lots of leadership books, but as I'll come on to in a minute, I'm an avid sports fan, and I used to play rugby when I was younger. And so one of my favorite books about leading things is called Legacy by a guy called James Kerr, and it's all about the All Blacks rugby team and their culture and how they create a sort of selfless environment where they all work for each other. And, they've had 140 years of unbelievable success, so they must be doing something right. Music. So I enjoy music, but I'm not an aficionado. Laura and I like to go to the symphony, but we also like other forms of music as well. And where I live in Michigan, there's actually a really lively music scene that we take advantage of. I probably shouldn't tell people this, but when I was a teenager, I was a punk rocker. As was one of my great colleagues, Debbie Law, actually, who I work with at Merck and BMS. We used to sometimes send each other little bits of music from iTunes. But my real passion has been around sports. I loved playing rugby. I played when I was a student at Oxford. And actually, I learned a lot of the things about leading people and working with a group of high-performing individuals in that environment. And we had, I think, 5 or 6 international players on the team I played with. They were much better than I was, but I was the captain of the team. And so I guess in a way it was a sort of forerunner of that situation where suddenly you're trying to lead something, but you have a group of people who can do that something better than you can. And how do you do that? So that was a huge privilege. And I've always been interested in the sort of way teams work and the dynamic of that.
More living it than reading about it.
Yeah, that's right. Exactly. My youngest son is a basketball player and in Division I basketball. And so he and I share a sort of profound interest in these things. It's an important bond between us in thinking about training and practicing and trying to get mentally right for things. So that's been a really important influence in my life.
Right. It's excellent. So perhaps we've looked back.
Yep.
We've looked back at your career. I think we talked about innovation and new products. When you look forward, thinking about the next 25, 50, 100 years, what do you see ahead in terms of healthcare, drug development, product development?
Well, probably most people would give the same answer, but I just can't help but be incredibly excited and optimistic about the application of large-scale data and artificial intelligence and machine learning to what we try to do. And all through my career, actually, it's been data. And it's been really important in improving things, I think, during that period of time. But it just feels to me like we're on the threshold or in the middle of a period here where it's going to improve exponentially.
Yeah.
I mean, the confluence of the compute power, the emerging skill sets of people coming out of our universities and into our industry, the technologies for creating and aggregating data. And already we can see some of the benefits of this. I mean, almost every company in Flagship is AI enabled. And they're already doing amazing things. I mean, like Generate Bioscience can, design an antibody on the computer, which is either better than any antibody you can find any other way, or can target epitopes that are almost impossible to raise an antibody to through conventional methods. I mean, that's amazing. And that's just the beginning of the things that we're going to be able to do. In BMS, we had implemented AI into all our small molecule drug discovery programs because we were working with you at Schrödinger on some of these applications too. So you have seen this. I mean, I've already seen examples of how this can not only accelerate programs and make them more efficient, which is the low-hanging fruit, but actually take you to chemistry that the human bias just won't find.
Exactly.
So I think we're just at the tip of the iceberg here. I mean, 50 years from now, unfortunately, I won't be here to see that, but I'm not even sure we can imagine
That's right.
what this will be like, because 25 years ago, like I said earlier, when I was seeing those patients in my exams, I mean, we had wards full of people with 90% psoriasis who could never go out of their homes and had to be covered in creams and treated with UV light just to be sort of barely functional.
Right.
Now you can have a 90% clearance from a monoclonal antibody. If you got melanoma, and it was beyond a certain Breslow thickness, that was it for you. Now people can be cured with immunotherapy. So those were things we couldn't imagine really then.
Yeah.
So 25 years from now, maybe we can sort of imagine some of those things. 50, if we continue to invest, and the need for this is huge because healthcare is unsustainably expensive and drug discovery is too expensive too. We know that.
Yeah.
The failures are burdening us. So if we can move up the probability of success by finding the right targets, finding the right populations, designing the best molecules, and then most important of all, I think running the right trials, which is where things get really expensive and we lose a lot of the signal, then I think it's really bright.
Yes.
Future.
Yeah. We don't have the answers. You mentioned that moment when you are able to run a good experiment and see whether you're right or wrong about a hypothesis. I feel as if our ability to ask more questions, whether it's in the lab, whether it's in the clinic, that's just going to explode, I think, because of the assist that we get from machine learning.
I think so. I mean, look, obviously there are worries about AI and machine learning in all sorts of aspects of life. Unfortunately, everything that we seem to come up with has a good side and a bad side. But, if applied in the right way to what we try to do, I think this could be amazing. Obviously, we've got to have the right data, but the ability to generate large quantities of data is also much better. And obviously, the ability to assemble information about patients and people is also much better. And that's improved in just the last few years, and it'll go on improving. So I'm optimistic about this. I feel like it's a really, if you were coming into the industry now as a sort of postdoc or somebody joining from a fellowship and you've got 25 or 30 years ahead of you to work here, this should be an amazing transformational period of time, I think.
Agreed. Yeah. And I guess we saw it that way thinking about molecular biology, being able to clone out targets and genes. But I do agree with you that I think there's going to be some amazing acceleration. We still have, 20,000 diseases that we haven't really made a dent in.
Right.
So while we've seen amazing progress with immunotherapy and, as you mentioned, cell therapy curing people, I think that this is just the beginning. Rupert, it's been absolutely terrific. Thank you so much. I've really enjoyed this.
Thanks, Karen. I appreciate it.